Hey everybody, this is the first of 4 lectures for muscle tissue histology. In this first lecture, we're going to be going over skeletal muscle, so the cells of skeletal muscle, and then the histological composition of skeletal muscle tissue. This video is sponsored by Doc2Doc, the personal lending platform designed for doctors by doctors. Do you have some big expenses in the near future? Maybe you're moving, applying to residency or fellowship, fixing up your car or home, or starting a new practice. Doc2Doc believes that traditional lenders overestimate the risk of lending money to doctors, residents, and medical students, focusing too much on the challenges of their financial past and giving them insufficient credit for the promise of their financial future. Check out Doc2Doc's personal loan options at doc2doclending.com/DaVinci. So first, just a general overview of muscle tissue. Again, muscle cells that utilize ATP and cytosolic calcium to power mechanical contraction. That's pretty consistent across all 3 types. 3 different types: skeletal muscle, which is mostly voluntary. So what we mean by that is, in your brain, you decide to move your arm, move your hand, move your leg, and skeletal muscle will respond and obviously move the part of the body that your brain is instructing it to. And that'll be via contractions producing movement. Then you have cardiac muscle, which is involuntary, and obviously it contracts to pump blood. And then you have smooth muscle, which is also involuntary, and you have contractions that narrow epithelial lumens, such as in blood vessels or the GI tract. So if you look at it, these 2 are involuntary. Skeletal muscle is mostly voluntary. One exception to that would be the diaphragm, which is skeletal muscle. And if you remember the diaphragm, if we draw kind of a crude drawing here of the thoracic cage, the diaphragm is this dome-shaped muscle like this, and it goes up and down during breathing to change the intrathoracic volume, to change the pressure gradient from the outside world, to drive movement of air in and out. And you'll learn more about that in your anatomy classes. Just a general histological point I want to make here. We'll go into the detail of the histological appearance of each of these later in the lectures. But as far as the big thing you want to remember is that skeletal and cardiac muscle are both striated. So what that means is that you have fibers composed of repeating contractile units called sarcomeres. We'll go over what sarcomeres are a little bit later in this lecture, but they're essentially the contractile unit of these 2 types of muscle. And they form this kind of striped pattern, this repeating band pattern. If you see here, it's a light band followed by a dark band, and that's a result of these fibers that comprise the sarcomeres. It's essentially there's a thin and thick overlapping filaments that produce these light and dark staining lines that you see on skeletal muscle and cardiac muscle. You can see them here as well. If you focus closely here, you can see the bands here. Now, if you look at smooth muscle, and part of the reason it's called smooth is because it doesn't have those striation appearance to it. And so the smooth muscle does not have that. And so they use a different contractile unit, and we'll go over that when we talk about smooth muscle in more detail. So now we'll go over the skeletal muscle structure. So we'll start over here. You have your muscle itself. So this could be, like your biceps muscle, your triceps muscle, deltoid muscle, any skeletal muscle in the body. So one thing to remember is skeletal muscle is highly organized. So it's organized first into the next layer would be muscle fascicles. And so down here you have a fascicle. So here's the entire skeletal muscle. Then you have a fascicle here. And then the fascicle is a functional unit composed of myofibers. Now that would be going one more down here. Now this is where things can get confusing. There's 3 different names for the same thing. So you have a muscle fiber, which are bundled together into muscle fascicles, which are then again further bundled together to form a muscle. And a muscle fiber can also be called a myofiber. And you can kind of remember that because myo refers to muscle, and then obviously it's a fiber, just like this. The other thing that gets confusing though is it's also considered a muscle cell. So fiber, sometimes we think of, a protein or something like that. In this case, it's actually a muscle cell. You got to remember that in this case, it equals a cell. So this is the muscle cell itself. And so these muscle cells are bundled together in essentially as fibers. And then they're bundled within these muscle fascicles, which are then bundled together within to form the entire skeletal muscle. And one thing to remember is that it's the reason why it's also called a muscle fiber, even though it's a cell, is it's a syncytium of cells, which basically is a group of cells fused together. So there's actually multiple nuclei. And again, we'll go over this in more detail when we go through each of these individually. And then these myofibers or muscle cells, they contain a series of myofibrils. And these myofibrils are a series of connected sarcomeres, which again is that contractile unit spanning the length of a myofiber or a muscle cell. So if you have, let's say this is your myofiber like this, you're going to have repeating units like this of sarcomeres across the entire length like this. And they're going to have these overlapping filaments like this, and this is how you get that striated appearance. And so each of these units is a sarcomere. And then lastly here, to explain a sarcomere, we'll go over a sarcomere in much more detail a little bit later in the lecture, but it's an overlap of thin and thick sliding myofilaments, which are essentially the filaments that slide back and forth to produce contraction, and thus, as we call it, the contractile unit. And so again, so you have the skeletal muscle, then it's organized into fascicles, which are bundles of muscle fibers or muscle cells, and then the muscle fibers or muscle cells are contained myofibrils, which are a series of connected sarcomeres. The other thing is that helps with the organization of skeletal muscles is the support of 3 connective tissue layers, and we've outlined these here in blue in this diagram. So you have the epimysium, which is dense, irregular connective tissue. Sometimes this can be a test question. You can get a slide or a diagram like this, and they'll point to the epimysium, or they'll reference the epimysium, and they'll ask you what type of tissue it is. So be aware of that. It's important to know what type of tissues they are. Really going forward in histology course, as we go through these basic tissue types, and at this point we're getting near the end of the basic tissue type unit, going forward, you need to know what different parts of different organs, what specific type of tissue they are, what specific type of epithelium, what specific type of connective tissue, and so on. So this is dense, irregular connective tissue surrounding all the fascicles that comprise the muscle. So this dark line outside here, that is the epimysium. So it surrounds it's essentially the case of the muscle. Then it goes further. You have the perimysium, which helps separate out these fascicles. It helps bundle all these myofibers together to form the fascicle. This again is dense, irregular connective tissue. Then you have the endomysium, which is a layer of reticular fibers encasing individual muscle fibers, which again are also known as muscle cells and myofibers. So now myofiber, which again, I'm just going to repeat this multiple times just to get this down. It's also known as muscle cell or muscle fiber. Key is, remember, these are cells. Okay, they're not proteins, they're muscle cells. And they're multinucleate syncytium that results from the fusion of hundreds of myoblasts. Myoblasts are embryonic precursors to muscle cells. So during development, hundreds of myoblasts will fuse together and they'll form a syncytium that becomes the muscle cell. So here, this is a cross section here of skeletal muscle. And you'll notice here that you have multiple nuclei within each cell. So this is a cell here, this is a cell here, obviously, and so on. And you see multiple nuclei. Each muscle cell is again surrounded by endomysium and then an external lamina with reticular fibers. One thing to remember is that myofibers or muscle cells are static cells, so they do not undergo cell division. They do not re-enter the cell cycle. They have exited the cell cycle and they don't go back. But the one difference, though, between, say, neurons or cardiac cells is skeletal muscle can more easily be regenerated by satellite stem cells that are within the external lamina. So although you can lose a skeletal muscle cell, you can actually regenerate new ones with stem cells that are present around. One thing you'll notice here, looking at this cross section especially, you can also see it down in this longitudinal, I'll show you in a second, is that by having a fusion of cells, you have multiple nuclei. These nuclei, which is hallmark for skeletal muscle, is they're located periphery. So here, like here's the center of the cell here. You see here's a nucleus here, here's a nucleus here, nucleus here. All these nuclei are peripherally located. They're actually just beneath the plasma membrane of the muscle, which is known as the sarcolemma. And so that's hallmark of skeletal muscle. That's one way you can tell that something is skeletal muscle on histological slides. So if you look at the longitudinal section here, one thing I'll point out that's kind of obvious is you can easily see the striations on a longitudinal section. Cross section, you can't really see it. So essentially the cross section would be as if you cut through here and then, obviously looked at it head on. So down here you have the cross section, and then you have multiple peripherally located nuclei. So you can see that here. Notice how they're not in the center of the fiber here. They're on the outside. Same thing here, and there's many of them, as you can see, and they're located just below this plasma membrane here. Muscle cells also have numerous mitochondria. They need a lot of energy to produce muscular contraction and muscular force. So it makes sense that they would be loaded with mitochondria. That's also what contributes to this eosinophilic appearance of the cells on H&E staining. And the other thing I'll point out is that you can't really see this on light microscopy, but they have specialized endoplasmic reticulum called the sarcoplasmic reticulum. And the big difference between the SR and the ER is that the SR has a higher, ER obviously contains calcium in any cell, but the sarcoplasmic reticulum contains a higher level of calcium, and that makes sense because calcium is needed for muscular contraction, which we'll go over later in the lecture. There's also other structural differences that we'll get into later in the lecture as well. So 2 topics I just want to touch on briefly are muscle hypertrophy and atrophy, and what's the difference. So muscle hypertrophy is an increase in muscle cell size. So it's just like any other type of hypertrophy, cardiac cell hypertrophy, any other type of cell hypertrophy. It's an increase in the size. Remember, hyperplasia is an increase in the number of cells. We went over this back in the cytology lecture. So remember, muscle cell or myofiber size. And that on the cellular level, what this is a result of is an increase in myofibril size and number. So you have an increase in the size of myofibrils and in the number of myofibrils. You also have an increase in glycogen stores. And so all these together contribute to increasing the cell size. This occurs in response to exercise and strength training. So this is how working out over a long period of time can increase muscle mass and increase muscle tone. And this is on the cellular level how this occurs is it's muscle hypertrophy. Muscle atrophy, on the other hand, is a decrease in muscle mass. You can often see this in very sick patients in the hospital or patients with certain neuromuscular diseases. And it's the opposite. It's a decrease in myofibril size and number. And it occurs in response to a number of different things. One would be just decreased use. So let's say you have a 20-year-old. It's not just very sick patients. Let's say you have a 20-year-old patient. They break their leg. They're in a cast for an extended period of time. They don't really move that leg. They're otherwise moving. They move their arms just fine. They walk with crutches, but they're not moving that broken leg. That leg is going to develop muscle atrophy because those muscles just aren't getting moved. It's essentially from different types of mechanisms, whether it's external or at the cellular level even, that prevent muscle movement. And so in that case, obviously the injury prevents muscle movement. You also have immobility. You see this a lot with patients who are in the hospital for long periods of time. It's one of many reasons why being in the hospital for a long period of time is not good for somebody in the long term. And so when someone's immobile, they're not moving their muscles aren't getting stimulated. And so that can happen. This is a particular concern for patients that are in the ICU. So the ICU, obviously, they're even more immobilized because they're usually very sick. And so, again, this is often a concern for ICU physicians. So cachexia is basically weight loss and muscle wasting, and you see it in significantly chronic diseases such as COPD, chronic obstructive pulmonary disease, heart failure, cancer. And these diseases, they result in poor nutrition, poor perfusion, obviously in the case of heart failure, poor oxygenation in the case of COPD. And so what happens is, and then cancer, obviously the cancer cells are very metabolically demanding. And so what happens is that other tissues suffer as a result of that. And so you're not getting adequate nutrients and perfusion in diseases like this to parts of the body, including the muscles, because the muscles demand a lot of oxygen, a lot of metabolic nutrients. And so as a result of that, you can see decreased muscle mass as a result of that. Certain medications like steroids can cause muscle atrophy, and then decreased neural stimulation. So this can be both at the central nervous system level and at the peripheral nervous system level. If you haven't had neuroanatomy, central nervous system is basically the brain and the spinal cord. Peripheral nervous system is everything else, essentially peripheral nerves. So like the median nerve, the axial nerve, sciatic nerve, you get the picture. So on the CNS side, let's say you have a stroke. And let's say as a result of that, you lose part of the brain that controls movement of your right arm. And so what happens with these patients is that since they've lost that neural input, the muscle doesn't get used. You can see there's a common theme here. If the muscle's not being used, either if it's not being used or it's not being perfused or has a high level of nutrients given to it, such as malnutrition, then the muscle will decrease in muscle mass. And so in this case with a stroke, you often see atrophy in the residual atrophy in the area that was the area of the body that was affected by the stroke. And so if you have a part of the stroke that affects the part of the brain that would, say, for example, the right arm, then you can see right arm muscle atrophy long term in these patients, unfortunately. Peripheral nervous system would be obviously at a very gross level. If you would cut a peripheral nerve, obviously it's going to take a while for that nerve to grow back if it ever does. And so you're going to have decreased neural stimulation as a result of that. It could also be nerve injury. The other thing is intrinsic muscle disease. So examples of this would be like ALS, Lou Gehrig's disease. So you have decreased stimulation of muscles in that regard. You could also have Duchenne muscular dystrophy, which we'll actually talk about at the end of this lecture. This is a pediatric disorder or a genetic disorder that results in muscle breakdown. And so as a result, we can have breakdown within the muscle as well that can lead to disuse and atrophy. So muscle fibers, they're actually classified into 3 different types. Remember, muscle fiber, myofiber, muscle cell, they're all 3 the same thing. So type 1, also known as red fibers, these are slower contraction and they're able to work for longer periods of time. So as a result of that, they contain high amounts of mitochondria, which contributes to that red appearance, and they rely much more on aerobic respiration, which is carried out by the mitochondria. That allows them to work for longer periods of time, and as a result of that, they utilize both fatty acids and keto bodies in addition to glucose. Glucose would be the main thing, but over time, as you run out of glucose, they can also use fatty acids and ketone bodies. They have a high myoglobin content, which is a specialized type of hemoglobin found in muscle cells, to help facilitate more efficient delivery of oxygen to the muscle cells and movement within muscle cells to the mitochondria specifically to facilitate aerobic respiration. And like we said here, they have numerous amounts of mitochondria. Type 2A, intermediate fibers. So these are faster contractions than the red fibers. They're still, though, even though they contract fast, they're fatigue resistant. These fibers, since they're slower contraction, they're able to work for longer periods of time. These are also considered fatigue resistant. Back to the intermediate fibers, they also contain high amounts of mitochondria and myoglobin for oxidative respiration. They also have large reserves of glycogen to help with as well. This helps with fatigue resistance, so you can keep driving this aerobic respiration with breaking down glycogen. They can also, however, switch to anaerobic respiration if needed. Type 2B fibers, also known as white fibers. These are named because of their appearance on histology. So these are fast contraction, even faster than intermediate fibers, but as a result of that, they're easily fatigued. This would be, say, sprinting, playing high-intensity sports like boxing, basketball, soccer, different sports like that, versus this would be more like, cross-country running. You could think of it like that. Or marathon. And so, this is obviously you need to have muscle contractions over longer periods of time, versus when you're sprinting, you need very fast contractions. These primarily rely on anaerobic respiration because it's just quicker via glycolysis and then glycogen breakdown to keep feeding that. They have low myoglobin content. That makes sense because they're not relying as much on aerobic respiration. They're relying more on anaerobic, which anaerobic means it doesn't need oxygen. They also have fewer mitochondria as well because, again, they're relying more on anaerobic respiration. Thank you so much for watching this video. Please like the video and subscribe to our channel to check out more histology videos and other medical education videos.